Just a little introduction for those who don't know who I am. Yes, I'm Lizzie, I work here at the CBU. Usually I'm interested in things about sensory control, particularly in the visual system, but today I'm going to talk about some work more on the method side of what I've been doing, combining non-invasive brain stimulation with neuroimaging methods. I'm based in Alex's lab, and just up front, I'm going to say I benefited hugely from a lot of very frustrating hands-on setup work from Jade, who literally just walked in the room. Hello, Jade. As well as Renhao, that you heard from this morning, and Katrina, who used to be at the CBU, I believe now is in Edinburgh. So thank you very much to them for all their hard work. I then took advantage of. Okay, so let's start from just saying that TMS is both a tool and a treatment possibility. So we can use TMS, which is a form of focal brain stimulation, to intervene with reasonably high temporal and spatial specificity in the human brain. And excitingly, it's now also being incorporated into the world of the clinic. So in the UK and several other countries, it's now approved as a treatment for persistent depression. So maybe where drug treatments or talking therapies have not been so effective. So it's a very exciting time for TMS. TMS, like any intervention, requires calibration. So we can talk about something like the dose of TMS, like we can talk about the dose of any other drug or medication you might be taking. We want to try and calibrate that to the individual. But there are several things with TMS we might have to take into account. So which particular machine are we using? Which coil are we using? So there's things on the technology side, there's things about your brain anatomy, and also some kind of more physiological measures. Are you actively doing a task or not? One really important parameter is the stimulation intensity. So how strong is the magnetic field that we generate when we deliver those pulses? So I'm going to call this stimulation intensity. We're going to treat it as a factor that contributes a lot to our kind of dose setting problem. So the kind of gold standard method for doing this is to collect something, measure something that we call the motor threshold. So to do a motor threshold, this is a nice picture I took from these people on the right-hand side. So we place our TMS coil on the head, and we might move that coil around until we start to see a visible twitch in your hand. You can also measure this with electrodes on your hands if you want a super precise measure. But what we typically find is that there's a hot spot on your head where if we deliver enough TMS, we will start to reliably see that visible twitch. And this is that level of intensity is what's referred to as your motor threshold. It's pretty stable across time, so we could do that 10 different times on 10 different days, and we would find within an individual it's pretty stable. So it's quite a good measure for calibrating TMS. However, often we're not stimulating the motor cortex, so do other parts of the brain act in the same way? When we stimulate other parts of the brain, obviously we're not expecting to see a hand twitch. And here's an example from the visual cortex. So when you do more posterior stimulation, some people report this experience of phosphenes. So imagine twinkly kind of lights or patches of motion. They're quite hard to describe, which is one of the problems with this method. But if you find people who can reliably report these phosphenes, you can get a phosphene threshold in the same way. How much TMS do we need to give you before you report this phenomenon? And there's some slight controversy over this, but in general, a few people find that it doesn't reliably correlate with your motor threshold, suggesting that actually these two brain regions require independent calibration. We can't just generalize what we know about the motor cortex when we stimulate other brain regions. So this might be one solution for the visual cortex, but again, we're not always stimulating visual cortex either, so how do we deal with that? Maybe we could take into account some anatomical considerations. We know that different brain regions are different distance from the scalp, so when we're stimulating from the outside of your head, that distance might matter. There was some work in the motor cortex to show that this was the case, so if you systematically offset the coil, motor threshold goes up, and there's this nice kind of linear relationship. So this work suggested that if you know the distance a brain region is from the scalp, you can adjust your motor threshold accordingly. And this figure on the right is kind of trying to make the point that you need to do this on an individual basis, because between individuals, it's not always, different brain regions aren't always reliably the same distance from the head. So the red patches here show you where that relationship is particularly variable. So all of this is to say that we might want to take into account some anatomical considerations on an individual level to help us set the TMS dose when we're not stimulating motor cortex and visual cortex as well. What could we do? Well, what I'm going to propose here is that we do something called E-field modelling. This takes into account a whole other bunch of information about the different properties of brain tissues that we know about from anatomical and physiological studies. And E-field modelling gives us an estimate of the electric field that's induced when we do TMS across the cortex. So can we use this to get some idea of how to adjust our motor threshold? So our very simple proposal at the end of all of this is to say that if we know the E field at your motor cortex and we know the E field at any other point in the brain, can we adjust your motor threshold by using that ratio? So this is our project. Does this method of adjustment help us set an appropriate dose for an individual? And then we want to validate this approach, obviously, and that's where the concurrent TMS fMRI has come in. So these plots just show you, this was our target region, so we're going for an intraparietal region here. And this is some examples of the plots you can get from E-field modelling. So this was our kind of motor coordinate, and then this was our more posterior parietal coordinate. You can actually see it's quite widespread, which if anyone knows anything about TMS, we can talk about that sometime. But we're just interested in the E field estimated within a small sphere around our target coordinate. How did our project actually work? So there were kind of three phases in our data collection. First of all, we asked participants to come for a slightly extended structural scan. So we collected the standard kind of T1 structural, we also collected T2 and DTI information. And all of this can be combined together in the E-field modelling. We use SimLibs software, if anyone is familiar with this process. After they'd been for their structural scan, we ran these E-field models, so one at motor cortex, one at our parietal site, calculated this ratio and set that as what we're going to call our adjustment. And then finally, participants came back for a second session in the scanner where we collected data from single pulse TMS. The TMS was set at 6 different intensities. So we had 3 that related to their motor threshold. So we stimulated them at their motor threshold, 110% of their motor threshold, or 40%. So I'm going to describe those as sub-threshold control conditions, 40%, where we don't think anything is really going to happen, and then 2 supra-threshold conditions, or threshold conditions, 100 and 110. And then we replicated that structure of 3 conditions for our adjusted condition. So overall, that gives us 6 TMS intensities. How do you do TMS in the scanner? Well, lots of different ways, but this is the way we chose to do it. So each line here represents one slice acquisition, and what we've done here is just introduce a gap in between the acquisition of volumes, and we deliver our pulse just after the offset of one volume. So this avoids artifacts from your TMS coil in the scanner. So we can just treat the data from here on as artifact-free. Okay, so what does our E-field modelling suggest that we should do? For this particular set of coordinates, our E-field modelling within our sample told us that we were understimulating absolutely everybody if we had used their motor threshold. So the average was about 15%. This plot here shows you the intensity for our 4 supra-threshold condition for motor, 100% for E-field adjusted, 100% motor 110, and E-field 110. So these blue bars are both, sorry, the blue data is both, they're both larger than the orange data, and the largest is our E110. You'll notice a lot of these are clustering quite high up here. We're actually hitting the maximum stimulator output for this device at 110 E-field adjusted. For anyone who's done any TMS, you might be thinking, wow, they were really going for it. Just remember that in the scanner environment, just because of the way the equipment has to be set up, the stimulator loses power. So it's not as intense as you might be familiar with standard TMS. But as a practical point, yes, we were hitting the maximum of what we could deliver with this setup in the 110 in many cases. Just to tell you that our numbers we had from this adjustment, our suggested adjustment values were correlated with adjustment suggested just by distance, so just calculating that scalp cortex difference. But our numbers were actually lower. So for our sample, the distance adjusted value suggested would have meant we would be increasing stimulation even more. And I've also included a plot here to say that there was no relationship between motor threshold and adjustment. So it's not the case that people with low motor thresholds needed more adjustment. We weren't kind of underestimating a subsection of our sample or anything like that. Okay, so I'm going to show you the main results after all that build-up on the next slide. So what I'm going to plot is the bold signal within our target IPS ROI for these 4 conditions. And the contrast we've done is the suprathreshold versus the relative 40% subthreshold condition. So you're going to see 4 sets of contrasts, 4 contrasts. So here we go. So this is our data at the target ROI. So what we see is that the highest bold signal, or highest bold signal change relative to those subthreshold conditions, is for our adjusted conditions only. So E100 and E110. We get significant change in bold, which was very nice to see. So basically, bold signal is going up after we deliver single pulse TMS, but only when we deliver those pulses at the adjusted intensities. And we also get a difference between the 110 and 100, as you might expect. I just want to put some caveats on this data immediately. So we also see a negative effect for our non-adjusted, our motor threshold conditions. I have to be completely honest, not sure exactly where this is coming from at the moment. Further investigations are ongoing. And we're also seeing that this is a surprisingly global effect. So we're not seeing a really tight local effect of TMS. Again, this is a bit of a work in progress, so any thoughts about that will be gratefully received at this point. But it's certainly saying at the moment that from what we've seen so far in the data, we get the most consistent increase in bold signal at our target site when we use this adjustment procedure. How am I doing for time? Got 1 more minute? Yeah. I put this in just in case anyone is interested in this method and this literature. There's been some debate about whether you can measure bold signal at the target site with TMS. And there was a recent paper showing that you can have multivariate effects, so we can decode the effect of TMS at the target site in the absence of a univariate effect. So they were suggesting that maybe the effect of TMS is a bit more distributed or a bit more not quite so consistent over voxels. We see exactly the opposite. So we can't decode it, but we do get this univariate effect. Maybe because of our design wasn't optimized for multivariate analysis in this case, but just in case anyone is interested in this kind of literature. We also looked at how our simulation parameters might have affected the adjustment values. So there's a lot of dots on this slide, but we looked at kind of 3 parameters that affect our modeling. There we go. So one is whether you model the E-field at the target ROI or at the cortical surface closest to that ROI. The other we've looked at here is, does it matter in your model how far away the coil is set from your head in your model? And the third is, how big does the size of the sphere you use to analyze your E-fields matter? And just to point out that the only one here that matters is the analysis radius. So you kind of exaggerate this adjustment effect if you take a really tight sphere around your target location, and if you have a big enough sphere, at this what's labelled here 20 millimeters, that effect kind of washes out. So for anyone interested in doing E-field modelling and anything like this, It seems to not matter a huge amount for some parameters, which particular settings you choose, but you might want to think about how large a region you're going to consider when you're doing that comparison between two regions. Okay, so to summarise then, we used E-field modelling to see if we can adjust the motor threshold on an individual level as a kind of alternative dosing strategy if you're not interested in targeting motor or visual cortex. Only our stimulation at adjusted intensities led to an increased bold signal, so this is a potential method for adjusting that motor threshold dose. There's lots and lots of open questions, a couple specific to this project that I've already mentioned, but thinking ahead to how we might use this in other contexts. Do we see the same effect in task-based contexts? And the big question, what about RTMS? So obviously this was a single pulse protocol. How does that translate to cases where we're delivering more than one pulse in succession? So that's it. Thank you.